Hello and welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson and today's topic is enzymes. So let's learn together. All right, let's talk about enzymes. So first off, we have to figure out what an enzyme is. I've mentioned before in previous videos that an enzyme is a type of protein. And they're essentially biological catalysts, and they're made of protein. And so a catalyst is any sort of substance that's going to speed up a chemical reaction. So anything that makes a reaction either faster or easier or both is a catalyst. And enzymes are just a specific type of catalyst. If we look at this chart over here, we can see that there's a red line and a blue line. And the red line would be the entire reaction without an enzyme. Now we notice it has this really big hill, and that's the amount of energy that would be required in order for this reaction to happen. So this is without an enzyme. It's going to take a lot more energy to get this started. I kind of think of it like a roller coaster. You know how you have to kind of go up with that chain, the clunk, 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 all the way to the top for those old-fashioned roller coasters, and then they drop you using gravity. That's without an enzyme. With an enzyme, that's kind of like the newer versions that use like gravity rails or magnets or something crazy that really helps you go a lot faster. And it's got a much lower hill here. So we don't have quite a huge hill. And with the enzyme, it's going to be much faster to do the same exact process. So that's the key here. Same process, but less activation energy. And activation energy is just that energy in the first hill, the energy you need for a reaction to be able to happen. So enzymes take that activation energy and make it much less. Now, there are many different types of enzymes. We're not going to worry about them right now. This is not something you have to memorize unless you are going to decide to be a biochemist, in which case my hat's off to you, but I'm not going to worry about it. Now, enzymes, they lower that activation energy, which we said is the amount of energy that you need for a reaction to happen in the first place. And we can see it again here. It's just lowering that first hill. I think of it as a hill on the graph. So if you have, another good explanation would be if you're a runner and you're trying to get to the bottom here where the product is, that's your finish line. It would take longer to go through this entire course here with the normal activation energy, whereas this would be much faster. You'd have a small hill to get there. This would take a lot longer to go up that really big hill. So think of it almost as a distance thing. The activation energy is just so much bigger without an enzyme in order to get to your finished product. Enzymes are very specific. This is something you'll hear any teacher say over and over again. Enzymes are super specific. They're only going to work with one type of substrate. Now, this could be multiple substrates that are the same kind coming together, but one enzyme equals one substrate. And we call this the lock and key model. And you can kind of think of it this way. Your front door has a lock on it, I would hope. If you don't have a lock, we have other questions. But you have a lock. You should only have one type of key that opens your front door, whether it's a code, if you have one code that opens the door, or a physical key that opens the door. But it should just be one of these things. You shouldn't have about 20 different keys that all open the same lock. That would be not very secure. So an enzyme works 1-to-1, one lock to one key, one enzyme to one substrate. And again, you can think of it like a lock and a key, but you can also visualize it like the enzyme. The active site is essentially that keyhole, and the substrate is like the key. So the enzyme's like our lock, substrate's like a key, and the active site is like the keyhole. So when they fit together, it forms an enzyme substrate complex. And that's going to allow us to have this chemical reaction occur. Now, if we think of it this way, pepsin is going to break down specific proteins. What happens if it mixes with carbohydrates? Pepsin is our enzyme. Now, we know that its job, based on this, is to break down proteins. So will it break down carbohydrates? No, it won't. Protein is like the key. So pepsin is that enzyme, and the protein is the key. If you have a carbohydrate key, it's not going to work because it's not specific to that enzyme. So if its job is to break down proteins, that is all it's going to do. That is its job, its role, it's done. It's not going to do other things. So to give us kind of a whole picture here, we're going to go through the 4 steps that are needed for an enzyme to work. The first step is that the enzyme and the substrate have to be in the same location. They can't be in completely different parts of the cell or of the cytoplasm because that just won't work. They have to connect in order for this process to happen. So the enzyme and the substrate are next to each other. The next thing that happens is that the substrate will bind to the active site on the enzyme. This is what I talked about when the key goes in the keyhole of the lock, which is the enzyme. So the active site, is going to fit the shape of the substrate. So they're going to match. Sometimes the active site will change to fit the substrate, but sometimes it won't. So typically they fit together really well, but sometimes it will mold to fit the shape of a specific substrate. And that's a conformational change. And it forms that enzyme substrate complex because they are together now. And we can see that here. We've got an active site, and notice this active site is differently shaped than this substrate here. So what happens is the substrate will enter the active site, and then the active site will change or do that conformational change, that shape change, in order to fit the substrate exactly. And that's what forms that enzyme substrate complex. So those are the first 2 steps. After that, the substrate molecule is changed. That means the chemical reaction happens. The whole point for this, the whole point for enzymes is to make a chemical reaction happen. So in step 3, once we have that enzyme substrate complex, this happens. The chemical reaction occurs. And then after that, we're done. The enzyme is going to release those products, the brand new molecule or molecules it's made, and it can get reused. That's one of the other nice things about enzymes. It is recycled and it can be used an unlimited number of times. If an enzyme eventually gets worn out, it can be broken down and rebuilt. So proteins are pretty hardy like that. You can rebuild them, you can break them apart and make new ones from different pieces, but proteins are pretty durable and these can be recycled. It's not a one-time use. And here's a full picture. I'm going to walk through the whole process one more time. We have over here our enzyme. It has an active site in it. The enzyme is like our lock, the active site is like the keyhole, and our substrates are like our keys. In this case, we had 2 substrates, and they came to bind to the active site, forming an enzyme substrate complex. We can see that here. We had 2 substrates. Now a chemical reaction is going to happen here, which we see the results of over here. Those 2 substrates have been combined into one single substrate, which is now called a product. So as soon as that chemical reaction is over, they're not substrates anymore, they're products now, because it has been produced by this process. And then after that product is released, the enzyme is unchanged, so it can be reused over and over again. Now, something can happen to our enzymes. They're not always 100% perfect. They don't always stay exactly the same. They can degrade. And this process is called denaturing or denaturation. Each enzyme has optimal conditions that it thrives in, kind of like us. If you throw me in 0-degree weather, I am not going to thrive. If you throw me in 120-degree weather, I am definitely not going to thrive. So similarly to that, enzymes like their ranges of things, like temperature and pH. Remember, pH is how acidic or basic a substance is on the pH scale. So our enzymes really like a specific hotspot so that they can work appropriately. If they kind of go outside these ranges, our enzyme gets damaged and typically changes shape. When it does that, the lock and the key cannot fit together anymore. Sometimes they can't fit together at all. And sometimes they can only partially fit together. So it may work only a little bit, or it may not work at all anymore. So denaturing is bad for your enzyme. It makes it so it will not work anymore or will work a lot less effectively. And this is what we're essentially looking at when we denature a protein. Here's our functional protein. It's all twisted up in very specific ways. And if you, let's say, heat it up too much, like you decided to cook it for some reason over like 140-degree heat, it would denature and unwind itself. It unravels into this denatured protein. It looks like a ribbon, which it's not going to work anymore. There's no way for anything to bind to it. It's just kind of a mess at that point. This is what happens when you cook eggs. So when you crack an egg into a bowl, you've got the yolk and you've got the clear part, which we call the white. Now, before you cook it, it's not white, it's clear. And that's because it's mainly protein. It's called albumin protein. Once you cook this egg and you expose it to lots of heat, that protein albumin is going to denature, because when holding something over a flame is not going to be a happy situation for an enzyme. It's going to be outside the normal range for that enzyme. So it's going to denature or get destroyed. That's how I kind of think of denaturing. It's like destroying the protein. So when we denature that albumin in the egg white, it turns white and it cooks. And this is why you want to cook things like chicken or eggs. You denature the proteins that could potentially cause you to get sick. So like certain proteins in bacteria, like salmonella, they are denatured and killed when we cook them. And that is why you cook chicken. Don't eat pink chicken. It needs to be white because you need to denature all those proteins. I'm going to kind of walk you through a couple of the graphs you may see regarding enzymes because they're very, very common, especially on quizzes or tests or just when you're looking to see if enzymes are working. They always operate best in their optimal conditions. So for example, we have a pH scale here from 0 to 10. 7 is neutral. So anywhere kind of right around the 6, that's going to be neutral. So right here, we've got our 7. That's neutral. If we get higher than 7, that's going to be basic on our pH scale. If we get lower than 7, that will be acidic on our pH scale. Now the pH scale does go higher than 10, but for our purposes, we're just looking at this graph. Our enzyme activity is increasing as we go up in this graph. So we have pretty much 0 enzyme activity at pH 0. And it slowly increases, and then it starts increasing a lot when we get between 6 and let's say 9 pH. Anywhere between 6 and 9 pH, we've got some pretty high enzyme activity, and it looks like it peaks right around 7. Right around neutral is where this enzyme is in the happiest, most active spot. If you expose this enzyme to a really basic substance or a really acidic substance, it may denature and be unable to work anymore. Something similar may happen with temperature. And again, this is a really good graph to show you this. So our temperature, this is in degrees Celsius. So for my American people, kind of fix your brain a little bit. We've got 10 degrees Celsius down here, which is pretty cool. And then as we increase, our activity increases as well. So rate of reaction would be the ability of our enzyme to work well. That increases until we get to the peak up here, which is the optimal rate for our enzyme. It's that sweet spot, the happy place for our enzyme. And that looks like it's right around 47 or 48 degrees Celsius would be the happy spot. Now, as soon as it hits that optimal rate, that's where you want your enzymes operating. Once that starts getting too high in temperature, your protein starts to denature, your enzyme starts to denature, and it's not going to work as well. And then if you get to a certain point, that protein enzyme is gone and it is not going to function at all. So it's really important to make sure enzymes are working in that correct spot because if you denature a protein, and especially if you denature an enzyme, it's going to be gone. You'll have to break it down and rebuild it again before it will work.
So let's recap what we learned today about enzymes. We know that they are proteins and that they act as biological catalysts. And that means that their job is to speed up a chemical reaction. They do this by lowering the activation energy that's needed for a reaction to happen. So essentially, it's just making it a lot easier for a reaction to occur in the first place. And here are a couple of steps that help it achieve this goal. The first part that has to occur is that our substrate and our enzyme have to be in the same location. They have to be near each other. When the substrate fuses with the active site of the enzyme and a chemical reaction occurs, that substrate or substrates are changed into a product or products. One of the most important things to remember about enzymes is that they are incredibly specific. In most cases, one specific substrate fits one specific active site. In addition, enzymes are greatly affected by several things: temperature and pH. If the temperature or pH are too high or are too low, the enzyme may not work properly. And at certain high temperatures or at certain pHs outside this range, the enzyme may denature meaning that the protein literally starts to unravel and degrade. If this happens, the enzyme can work less well or not at all. Well, thank you for watching today. Please like, subscribe, and comment for more biology videos and content. And until next time, I hope you had fun, I hope you learned something, and I'll see you later.